Today we will discuss about Cachexia network changes in the brain. So far from the previous results we have understood that we can clearly distinguish the Cachexia, pre-Cachexia and the control conditions in the brain that we have discussed in this figure 2 where we have discussed the from the brains we can clearly distinguish between three different populations: control, pre-Cachectic conditions and Cachectic conditions. So they wanted to see what are the changes that occurs in the brains in the pre-Cachectic to Cachectic conditions. For this they did the C-FOCS uh, workflow screening, where they will analyze the neuron activations in the brain cells at different conditions. This in the control conditions, Cachexia conditions, and the pre-Cachexia conditions. So once the Cachexia is inducted, then the brain cells are immunostate and cleared, and it did the light sheet imaging. Then the cell detections and their registration in the atlas is done, and in this they are able to see that some of the genes are highly upregulated from pre-Cachectic to Cachectic conditions, highly downregulated. Around 41% of the genes are downregulated, and around 12 genes approximately, I'm not able to recall the exact number, 12 genes are upregulated in this pre-Cachectic to Cachectic conditions. And some of the spots are shown here, that is NAC spots and this PBIE are and the ARPs, and these three are highly upregulated. When they quantified these things in the cells, how many cells are have these activation signals, they are able to see that PBIE has the highest cell counts. And in terms of the down regulations, they see that AOD, PIR, VIS as the down regulated cells. And when they did the quantifications to find out whether this increase is statistically significant between the pre-Cachectic to Cachectic conditions, they are able to see that MNAC, PBIE and ARP, that is shown in the figure B, has the statistically significant upregulations. And when they did the PCA plot analysis of the pre-Cachectic and the Cachectic conditions, they can clearly distinguish between the samples that are from pre-Cachectic and the samples that are from Cachectic conditions. Now, apart among this PBIE, MNAC and ARP, which one will cause highest disruptions or network changes in the brain? For that they did this studies with different combinations, that is PBIE alone, PBIE plus BRTS, PBIE plus PBHM and PBIE plus MNT spots, and they are able to see that the highest disruptions are usually caused by this PBIE, which is more sensitive to us to the network changes in the brain. Then they did the heat plot map analysis, and in this case what they are able to find out, they are able to see a clear changes in the genes from this that are shown in the bottom side of this PBIE, and we can see that they are highly upregulated in the initial regions, and then they are downregulated in the later group set. So this downregulations is mainly related to this physical activities of the mice, and whereas the upregulations are mainly related to the essential activities that is necessary for the survivals. So this their bioinformatic analysis of this f-cos stainings indicates that some of the genes that are highly upregulated, and those might cause this network, and those might be the result of this network changes in the brain when the brain undergoes from pre-Cachectic to Cachectic conditions. And the genes that are highly downregulated, around 41% of the genes are highly downregulated. Those lists are not shown here, but their analysis are shown in this. Their analysis or results are shown in the supplemental figures. And here just some of the main cells that has an impact on these network changes in the brain are shown. If you need to understand more about their experimental results in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.